So what I want to talk about today is to try to synthesize a lot of what's been coming out very recently and very rapidly on SARS-CoV-2, and try to sort of provide some sort of reasonable background on a set of key questions on SARS-CoV-2 biology with an emphasis on entry and immunity. And so what I'm going to talk about today is a little bit of sort of general background on what makes a pandemic virus, talking a little bit about SARS-CoV-2 contextually with other similar beta coronaviruses, how it enters and infects cells, particularly thinking about the respiratory tract, and finally talking about what we know and what we might not know but have some beliefs about vis-a-vis immunity, and which of those might or might not be well supported. Also, just again, if someone has a hot mic, that's bringing some background noise through, so please again check that you're muted. There have been a number of comparisons to previous pandemics. People have estimated mortality. Most of these, again, are estimates due to large-scale infections in the past, and SARS-CoV-2 is certainly not the first of these. It's probably not going to be, we hope, the highest mortality of these. But there are a number of pandemic infections, viral and otherwise, that have swept large portions of the world through recorded history. There are several characteristics that I'd loosely say are particular to pandemics and pandemic viruses. One, obviously, is global spread. In thinking about SARS-CoV-2, we often talk about modern air travel and the effects of that. I would use this figure from a 1991 paper on influenza spread to simply remind everyone that global spread, while it's facilitated by modern travel, does not require air transportation. And indeed, in the 1918-1919 influenza pandemic that people refer to as Spanish flu, that there was global spread over a period of about a year and a half. The second key feature of a pandemic virus is the lack of prior immunity in a large fraction of the population, and that can result from new geographic range. Zika has thus far not ended up being a pandemic virus, but caused a large-scale epidemic spread, and that was in part driven by a new geographic range. Also, new host range, viruses that adapt to hosts other than the ones they've been circulating in. And so we've seen that with beta coronaviruses, and we see that with SARS-CoV-2. We've also seen this with avian influenza transmitting efficiently in humans, as in the 1918-1919 H1N1. And then also it can happen, also as illustrated by influenza, it can happen with new genetic material if there are recombination events or similar that introduce substantially new genetic material into a virus that has already been circulating in human hosts and make it different enough. Influenza is perhaps the prototypical case for studying and for risk assessment for pandemics, because it's caused a number of pandemic infections across history. And shown in the lower right here, the US CDC goes through a regular structured exercise trying to assess and score the emergence risk and the impact risk of different influenza strains that are not currently broadly circulating in humans, but pose some pandemic risk. And so those are the sorts of risk assessment exercises that public health authorities across the world use on a regular basis to try to look at some of these factors in pandemic spread. One of the things that doesn't necessarily make a pandemic virus is case fatality rate. And I think Ebola is a good example of that we've had large-scale epidemic spread of Ebola, but the high near-term case fatality rate of Ebola combined with the relatively early onset of symptoms compared to transmissibility actually works against its potential for large-scale spread. And you can argue that both influenza, SARS-CoV-2, and viruses such as HIV that have a large infectious period when they're not substantially symptomatic have characteristics that are better suited to the sort of spread that we're seeing today. So, sorry, I'll just back up one slide. So the ways in which SARS-CoV-2 sort of fulfills these is that it's a virus that is substantially new to human hosts, and therefore there is not much prior existing immunity. It appeared to have, it's most closely related to viruses that were circulating in bats, and so therefore appears to have made some sort of host range jump that underlies its emergence. And then it also has a substantial pre-symptomatic and potentially also asymptomatic transmission window that makes it particularly difficult to impose sort of trivial symptom-based control measures, and indeed the sorts of care-seeking behavior and self-isolation that people who are sick normally perform don't protect as much against the spread of the virus in this case. SARS-CoV-2 is genetically related to a number of other coronaviruses. Coronaviruses are single-stranded positive sense RNA enveloped viruses, and perhaps most distant, there are human seasonal coronaviruses that cause some of the symptoms that we identify as common cold viruses. These are not the only ones that do that, but they cause periodic infection in humans and are usually not terribly serious. There are a large number of other coronaviruses that are phylogenetically related, primarily not isolated from humans, many of these from bats. In the early 2000s, SARS was identified as a virus that had made a species jump into humans, transmitted among a number of humans very rapidly, causing a lot of deaths, but then ceased to transmit. And that's actually the closest phylogenetic relative of SARS-CoV-2 that circulates or has circulated in humans. MERS is another coronavirus. Both SARS and MERS, the ultimate native hosts are believed to be bats. MERS can also infect dromedary camels, and it causes disease primarily in the Middle East. It is more distantly related, as you see, to SARS-CoV-2, but still much more closely than the human coronaviruses. And then there are a lot of closely related bat coronaviruses that have not previously been seen in humans. So that's a sort of very brief genetic overview of this. There's been a lot of phylogenetic analysis by a number of top groups around the world. Looking a little bit more closely at SARS-CoV-2 and its sort of closest human infecting neighbors. These are schematics, a schematic and an electron micrograph for SARS. Both of these look very similar for MERS or SARS-CoV-2, but they're relatively straightforward envelope viruses with a spike protein that is responsible for receptor binding and, we believe, entry. a single-stranded RNA genome. The coronavirus name comes from this crown-like appearance of the spike proteins on the electron micrograph. And all three of these viruses cause a disease spectrum that can vary from asymptomatic infection to acute respiratory distress syndrome. The distribution of severity across these viruses varies between MERS, SARS, and SARS-CoV-2. So these are not all the same set of symptoms, but the spectrum of symptoms is highly overlapping across these. SARS and MERS primarily enter cells via the endocytic pathway. This is work that and some of the major contributors were DeHaan, Bates, Gary Whittaker, Judith White, and their co-workers. SARS-CoV-2, as well as these other viruses, as far as we're concerned, primarily infect cells of the respiratory tract. There's a question, and I hedge this by saying primarily, because in SARS-CoV-2, RNA sampling has shown substantial fecal shedding of RNA. It's not yet known if this is infectious virus or not. That can occur for a large period of time after infection. And this brings to mind, the reason I have a duck here, is a lot of work on avian influenza, where low pathogenicity avian influenza is shed fecally by ducks and is primarily spread via fecal oral transmission in wild waterfowl. However, high pathogenicity avian influenza like H5N1 infects the upper respiratory tract of the ducks and may be spread by multiple means. And so while we've very much focused on the respiratory tract for SARS-CoV-2, both because that's the major location of pathology and the way that we want to prevent death, this fecal shedding has raised questions as to whether there's the potential for transmission and especially extended transmissibility via fecal shedding. This has not yet been answered. So that said, a lot of the early immunohistopathology shown here shows immunostaining in lung sections. There's severe lower respiratory tract infection. There's also virus recovered from swabs of upper respiratory tract, and we believe also infection of upper respiratory tract cells. Although the precise range of cells and their role has not yet fully been elucidated. This is obviously a virus that is moving quickly, and both the clinical and the basic data are being accrued as we go. But the virus does infect lower respiratory tract cells and causes substantial alveolar pathology. The receptor for SARS and also SARS-CoV-2 is ACE2, and that is well expressed on the surface of lower respiratory tract epithelia. This has been demonstrated by a number of groups recently. There was a very nice paper by Michael Letko, but I'm showing a somewhat complex figure from at the bottom, where SARS and then also SARS-CoV-2 enter via ACE2. MERS enters via DPP4 and not ACE2, and a number of the other related beta coronaviruses enter via neither of these receptors. So there's some uncertainty for a number of related coronaviruses, but SARS, SARS-CoV-2, and the most closely related viruses all enter via ACE2. There was recently a liganded cryo-EM structure of SARS-CoV-2 with ACE2. I'm showing a rendering of the substantially similar structure of SARS in complex with these two in the upper right. So the structural, cell biological, and biochemical evidence are relatively in alignment for ACE2 as the receptor for binding. Activation of these viruses for entry appears to be primarily proteolytic, and this has been the case for a number of these other coronaviruses. Again, people including Judy and Gary Whitaker have done a lot of work on this for earlier coronaviruses. As best we understand for SARS-CoV-2, this is still work where we're accumulating evidence. Furin plays a role in proteolytic activation, but not necessarily cleavage, and this may happen on the way out of an infected cell. We're still figuring this out, but there's biochemical evidence for furin cleavage, and then activation and triggering on the target cell appears to happen via perhaps alternately two different sets of proteases. TMPRSS2 is a cell surface protease expressed on lung epithelial cells, and cathepsins, BNL, are endocytic proteases. The cathepsins are heavily implicated in SARS entry in the endosomes, and Judy's work again has helped lay that out. There was some very nice work that came out in Cell within the first part of this year on SARS-CoV-2, showing that inhibition of SARS-CoV-2, so inhibiting the cathepsins with E64D has a moderate effect on both SARS and SARS-CoV-2, inhibiting TMPRSS2 via chemistat has a more substantial effect, and then inhibiting both really drops the entry of both SARS and SARS-CoV-2. And so figuring out exactly what the alternative usage of these two pathways is, of these two activation proteases is something that is still a work in progress, and this may also very much depend on cell type. And the biochemical evidence suggests that TMPRSS2 may be particularly important in the lower respiratory tract, and therefore in the primary pathology of SARS-CoV-2. This Hoffman paper also looked at the effect of ammonium chloride treatment, which you see on the lower right, in a couple different cell lines, very much inhibited entry of SARS and SARS-CoV-2, although the precise magnitude of the effect does vary by cell type, again suggesting some variation in protease usage. So this is sort of my best attempt to make a coherent picture of data that are very much emerging at this time. So we have a good receptor. We know several proteolytic activation factors. The variation by the cell type remains unclear. The entry compartment, whether it is indeed endocytic, as for SARS, and whether it's early or late endosomes, or whether entry is in fact possible at the cell surface, is not yet well worked out. And also we don't know yet which tissues are commonly infected first and which are most responsible for spread. Again, by analogy to other viruses, especially work on various sorts of influenza, we would expect that upper respiratory tract tissues are likely infected first and are likely most involved in droplet spread, and lower respiratory tract tissues are most involved in pathology. But this is really speculation by analogy rather than hard data at this point. So shifting gears a little bit, I want to talk some about what we know and what we believe about immunity to SARS-CoV-2. And I'll say there are a lot of data coming out. There are also a lot of moving parts in antiviral immunity, and many of the other people on this Zoom conference are much more expert in many of those parts than I am. But both innate and adaptive responses are broadly involved in antiviral immunity. There are key roles played by both cellular and humoral immunity, and also different antibody isotypes. Types, that's a typo. That, classically, and indeed we see this in the serology of SARS-CoV-2 infection, that IgM comes up first as part of the acute response between 6 and 12 days after symptom onset, followed by IgG. However, I would remind people that this is a virus that infects via mucosal surfaces, and therefore the role of IgA may be particularly important. That's not something that's been investigated as much in the initial reports. And there's also some evidence from influenza that IgA may be particularly involved in neutralizing immunity at the time of initial infection, and also the differences in IgA neutralization versus IgG neutralization may help explain some of the differences in antigenic evolution of influenza. And so this may come to be very important as we start thinking about the change over time for SARS-CoV-2 and the role that human immunity may or may not play in that. There have been a few small patient series published with detailed immunologic data. I'm showing here some data on cellular responses in the N=1 patient series that was a correspondence to Nature Medicine, published recently, but again showing that CD4 and CD8 responses come up as you'd expect. Also there's some role for monocytes, and they have some more detailed flow data that I'd encourage you to go look at if this is of particular interest to you. One big question is to what extent can humans generate protective immunity against beta coronaviruses? That human seasonal coronaviruses people can be reinfected with over the course of their lifetimes. And we're not certain whether that's due to a loss of immunity with time or antigenic variation on the part of those coronaviruses. There's been more work done, obviously, for MERS and SARS than SARS-CoV-2, and we've had longer periods of time to do this. But these are key questions as we think about both the what natural infection will do in terms of protective immunity, and what vaccines and immunotherapeutics may be able to confer. So for MERS and SARS, perhaps our best data are in non-human primates. I'm showing here first one MERS vaccine study in non-human primates that suggested partial protection, where this was using a vaccinia vector, and as you see there, that the viral load was reduced somewhat with both high and low dose vaccines, perhaps not completely in a dose-dependent fashion and not as cleanly as one might like, but that there is some degree of protection provided in this study. There was another study for a MERS DNA vaccine in macaques, and this showed a nice reduction of lung pathology and reduction of viral load with this particular vaccine, again, suggesting the ability to provide some degree of protection, whether absolute or relative, with the vaccine. There's also been work done in dromedary camels, which again can be infected with MERS, suggesting that at least the camels are resistant to reinfection, that they do not show symptoms of illness upon reinfection. So again, suggesting that whether or not there might be some viral replication upon re-exposure, that there's some hope that for some of these related viruses, illness would be either absent or not severe upon reinfection. There's a paper that's been up on bioRxiv for about 3 or 4 weeks now and was just accepted at Nature. That is a series of 9 patients in Germany, followed all with mild to moderate infection, who were who acquired SARS-CoV-2, and both their viral load and serologies were followed over time. And so you see in somewhat small print on the upper left their viral loads via serial sputum swab and stool sampling, and also the orange arrows that you see seroconversion in most of these patients, but the point of seroconversion does not correlate particularly with the resolution of symptoms, and also does not strongly correlate with the reduction in viral titers. So the good outcome is that these patients were developing antibodies, but there's not a very simple, straightforward relationship between IgG and IgM titers and drop in viral titers. So this is an interesting paper to look at and to consider, both in terms of vaccine protectiveness and natural immunity, as well as the question of convalescent plasma and its potential therapeutic efficacy, which I think is something that I hope we'll have data on soon. There are a large number of people working on that. So also we're starting to acquire data on different immunoglobulins against different epitopes on SARS-CoV-2. And I show here one recent paper that propagated and characterized a large number of patient-derived monoclonal antibodies, particularly looking at monoclonals against the SARS-CoV-2 receptor binding domain. So the receptor binding domain is of course what's responsible for binding the ACE2 receptor, and in this study the affinity for the receptor binding domain largely, at least sort of block correlated with the neutralization titers against SARS-CoV-2. There are also antibodies against the NP, as well as antibodies against other parts of the spike protein. One thing that was noticed in this particular study is that there was not very good cross-reactivity in the receptor binding domain specific antibodies. And so there's also been work suggesting that some anti-SARS monoclonals can neutralize SARS-CoV-2, but not all. And trying to characterize a little bit better neutralizing efficacy, cross-protective efficacy, and then ultimately protection against challenge will be important questions going forward. One other note as we think about potential hopes and pitfalls for vaccines and for immunotherapy, I credit Akiko, sorry, Akiko Iwasaki for bringing this to the attention of the community, that there was a study, another vaccine study of SARS, so not SARS-CoV-2, in 2019 in rhesus macaques, where the macaques were given either a SARS vaccine vector or a blank vaccine vector. And again, I believe this is a vaccinia backbone. And what they saw was with the SARS vaccine vector, that's denoted ADS here, that there was a substantial reduction in viral titers, especially plot. You can see this very nicely in C here, in panel C, that the viral titers reduced very quickly after infection. However, there was substantial immunopathology in the lung. And you can see the histopathology in panel D, that the ADC-MVA looks like relatively normal lung tissue, maybe with a little bit more infiltrate than you'd like, but that the macaques that received the SARS-CoV vaccine had very substantial lung immunopathology, and they traced this to monocytic infiltrates that appeared to be mediated by the Fc gamma receptor engagement. And so this may be a way in which IgG-based immunity, at least in this particular study and for SARS, can play a role in substantial immunopathology. And that's also of relevance here because there's a question for these viruses of how much of the poor outcomes are direct viral lung pathology and how much are immune-mediated pathology in the lung following severe infection. So this is a potential pitfall and something that we'll all be watching very closely as time goes on. There have also been, and I haven't been able to get really good data, but you'll see in the media and various reports of people who have, quote, been reinfected with SARS-CoV-2, and following these fairly closely, that there are often patients who tested RNA positive for SARS-CoV-2 and then ultimately recovered, tested RNA negative, were released from the hospital, and then at some point later, often on routine surveillance, tested RNA positive again. There have been 1 or 2 reports where there were some symptoms associated with the second positive test. In most of these patients did not experience any symptoms at the time that they tested positive, and this was essentially an incidental positive test. And so I think there are some questions that this raises, both for the biology of infection and obviously for infection control and spread. Some of this may be simply a question of prolonged intermittent shedding, that this has been documented for SARS-CoV-2. This is the reports I'm referring to are all adult patients, but prolonged shedding has been particularly documented for children who often have mild or asymptomatic infection with SARS-CoV-2, but can shed at least RNA for quite a long period of time. And so this may be just prolonged shedding. It may be some sort of intermittent recovery of RNA and or infectious virus from tissues, or it may, and most people in the field think this is less likely, indeed represent a bona fide reinfection event. But I think in the absence of clear data for reinfection, one would tend to favor one of these other 2 scenarios. Those other 2 scenarios would, and even a reinfection with milder asymptomatic illness would argue for protective immunity, and so this is good for people not getting severely ill twice. It's also bodes well for a vaccine potentially if it could avoid these potential immunohistopathology issues. However, this potentially poses problems for sort of purely symptomatic or public health non-molecular infection control measures, that if people who recover can potentially shed not just RNA but infectious virus at later points in time, then this is a challenge for being able to say who poses an infection risk and who does not. Some questions that are people tend to pose in the field that I think we really don't have answers to right now are whether SARS-CoV-2 will become a persistent seasonal virus, such as some other human coronaviruses or such as seasonal flu. We simply don't know that. I think two questions there are whether mutation will allow it to ultimately evade any protective immunity, and how severe illness would be when people are infected with a drifted strain, and also how long that would take to happen. Also related to this is if people do develop protective immunity, how long will this last? That a number of the studies looking at long-term antibody titers in particularly non-human primates exposed to SARS and MERS show a decline in titers over time, over a small number of months. Whether that decline in titers correlates with a decline in protection or not, we don't know. That certainly when people are immunized against influenza, that we do see a decline in antibody titers over a period of months, but at the same time, the protection does last over even year-to-year time frame. So we don't know the duration of immunity to these viruses. These will be key questions to answer going forward. Also, a lot of people talk about herd immunity, and herd immunity is a concept that's really developed in terms of thinking about universal vaccination campaigns and thinking about infection control for things like measles and chickenpox and viruses that we have vaccines for that are recommended for universal uptake, and where the vaccine may not be 100% efficacious, and or there may be people who are immunocompromised and therefore vulnerable. But if a substantial fraction of the population is immunized and therefore cannot be infected and cannot transmit the virus, that we can protect those vulnerable individuals. And again, I would credit Akiko Iwasaki for this rendering, and also for a lot of discussion that this is something that's these are concepts that are very familiar to the immunologic community, but that I think are being used in an imprecise fashion by both some people in the political sphere trying to talk about public health decisions. That in the case of a virus such as SARS-CoV-2, if we're talking about mass infection of a large number of a large proportion of the population, that protecting vulnerable individuals via a herd immunity in the absence of a vaccine becomes extremely problematic. That those individuals would have to be sheltered for the entire duration of the large-scale infection. The infection would then have to result in neutralizing immunity, that the recovered individuals would have to not shed virus after they've recovered. Again, at least at an RNA level, we know that is not the case for children and for a number of adults. And again, that these recovered individuals would have to be protected from reinfection. I would also note that in addition to immunocompromised individuals, elderly individuals, and individuals with a number of comorbidities, that that healthcare workers are at increased risk for poor outcomes with SARS-CoV-2 infection. And I think it's simply not feasible to suggest that all healthcare workers should self-isolate for the period of a pandemic. So I think there are a number of fallacies in this application of herd immunity to the case of a SARS-CoV-2 pandemic, barring a broadly adopted vaccine. And I think a large number of people in the public health community and those making decisions about this have indeed turned away from that idea. But it's something that persists in some of the popular imagination. And it's important that as scientists, we be able to understand that and explain why it's not the case. So that's pretty much what I wanted to cover. I think there's sort of three key sets of points here are that SARS-CoV-2 has been able to spread rapidly and globally because of its infectiousness, pre-symptomatic transmission, and lack of prior immunity. Cell entry, as best we understand right now, is via binding to ACE2, proteolytic activation, first by furin and then triggering by TMPRSS2 and or cathepsins. The precise entry compartments and requirements are currently being determined. And immunity that blocks receptor attachment appears to neutralize the virus based on data for SARS. There's a potential for immunopathology. There's a potential for prolonged shedding, and there are a number of complications as we examine protective immunity. So thank you for your attention. I'd like to thank a number of colleagues and collaborators, some groups listed here that we're working with a large number of people, and in general, the scientific community, has really come together as we try to work together to offer what help we can against this threat. And I'm lucky to be collaborating with a number of wonderful groups and scientists at UVA in Sweden and across the world. And I'd like to thank in particular in my group Anjali Senkar for her hard work, Judy White as a valued colleague who always offers wise counsel, and both the Newt and Elise Wallenberg Foundation and the Commonwealth Health Research Board for funding, and especially to all the clinical care providers who are caring for patients with SARS-CoV-2 every day, and all of the people sharing data rapidly as we try to adapt and defeat this. So thank you, and I know there are a lot of people on the Zoom, but I'd be happy to try to answer questions as time permits.